Svendoli. He graduated in biology at the University of Kaiserslautern, and then he moved to the Institute of Human Genetics in Saarland in Hamburg, where he carried out his postdoctoral studies, not the same university. Then he became a group leader and assistant professor at the Department of Clinical Chemistry and Pathobiochemistry in Aachen, where I also was. And in 2004, since 2004, he is now full professor in Heidelberg University and head of the section of molecular hepatology and alcohol-associated diseases. So thank you very much for being with us, and please, whenever you want.
Professor Banares, also thank you for having me. It's a pleasure to Madrid and give a presentation about the topic. We have shown here TGF-beta signaling regulation and outcome in alcoholic liver disease. So I want to start with this scheme. I want to start with that scheme that shows how alcohol affects the liver. And actually there is a direct and an indirect route how alcohol can affect the liver. One thing is the direct effect that the alcohol molecule has on liver cells. It induces It induces steatosis and damage of hepatocytes. And based on that, you have cell death, you have damage in the liver, which then induces these different steps that we all know that are advance of fibrosis and hepatitis. This is inflammation, this is stellate cell activation and proliferation. This is stellate cell activation, which you can see here. And then we have the process of fibrogenesis that is induced by alcohol. We know that it takes time until we are there. It can be decades in the human situation. And that's also the reason why it is so difficult to set up animal models where we can comprime the whole procedure in a few months. We are more successful with the known models like CCL4 and BDL. And then besides this, we have the indirect route, how alcohol can affect the liver. And this is done by the gut. So here alcohol consumption leads to a so-called leaky gut. So the fenestration of the epithelial cells of the gut, they are becoming leaky, they open up, and then you have an increased flux from the gut inside to the portal vein. And in addition, we also know that alcohol is inducing the bacteria to proliferate and to increase the levels of endotoxins. And the net effect of this is that the so-called pathology-associated molecular patterns or endotoxins, how they have been traditionally named, they then affect the liver. They bind to the TLR receptors and activate Kupffer cells, activate stellate cells, and then on the indirect route, they can induce then inflammation and can enhance the damage situation that we anyway have already with the alcohol molecules itself. So there's a strong connection between the gut and the liver. So coming to TGF-beta, TGF-beta is known as a driver and a very important cytokine in the liver. So these are the different cell types that can induce or can produce TGF-beta in the liver. And we know that in a normal liver, you barely can find activated TGF-beta around, and the amount of TGF-beta is increasing with severity of liver damage. I don't want to talk today about what we know since long, that TGF-beta is a driver of stellate cell activation. So the active TGF-beta leads the transdifferentiation of quiescent stellate cells, which are dedicated to fat storing, to vitamin A storage, to the activated myofibroblasts, which are finally the cells that produce the extracellular matrix. I also will not talk about the TGF-beta role in the inflammation system. We know it's an anti-inflammatory cytokine, and it acts on macrophages, and it acts also on T cells and induces differentiation to regulatory T cells, which then act anti-inflammatory. We also know that TGF-beta has a pro-angiogenic role, and this is also not the topic today. So since a decade now, we have been focused on investigating how TGF-beta affects the parenchymal cells, in this regard, the hepatocytes. And here we know that there is some kind of switch behavior. TGF-beta has a so-called double-edged role. That means, in principle, in the healthy liver or in the proliferative situation, TGF-beta is cytostatic. It can induce apoptosis. It can inhibit proliferation of the cells. But at some point, it becomes a tumor promoter. And in this setting, the liver is also special. We know from other tissues like colon that the TGF-beta pathway is disturbed by mutation of some components of the pathway, and therewith then the cytostatic branch is just lost by mutation of SMAD proteins or by mutations of the type 2 receptor, for example. But in the liver it's different. So you rarely find mutations or changes in the components of the signaling pathway. And here we have a situation that this downstream signaling is just branched into a different direction. So we have still a SMAD activation, but here we have maybe other components that interact with the SMAD proteins, and therewith the outcome in the cells is different. I will come back to that later. So we should, at that stage, we should take that usually in a healthy liver or in a chronically, in an acutely damaged liver, TGF-beta is cytostatic, and in precancerous stages and in late stages of liver damage, it becomes a tumor promoter. With regard to alcohol and the effect of alcohol and the TGF-beta system in alcohol-mediated liver damage, we are currently breeding these mice, and I just have filed an application that we will be able to use these mice, which have then a type 2 receptor deficiency, when we use the respective CRE mice in the different cell types of the liver. So we want to delineate the role of TGF-beta signaling in a setting of the NIAAA alcohol feeding model with respect to liver damage and progression of disease. So a few words regarding to the TGF-beta signaling pathway. This is the canonical pathway shown here. So the ligands bind to the type 2 receptor, which then recruits the type 1 receptor or ALK5 by phosphorylation events. Then this complex is recruiting the intracellular mediators, the SMAD proteins or the receptor SMAD proteins, 2 and 3 mainly, and those interact with the common SMAD and then form a transcriptionally active complex, which is then translocated to the nucleus and with a battery of cofactors then dedicates the outcome and the target genes that are then induced or inhibited. The TGF-beta pathway is druggable at different levels. So that means we can interfere, we can use soluble TGF-beta receptors, we can use neutralizing antibodies. We have compound inhibitors, which are already also in clinical studies, especially liver cancer studies, which act at the activated type 1 receptor. We have components that interfere with the latent TGF-beta. I'll come back to that a little later also. So we have different possibilities to interfere with the TGF-beta signaling pathway. And what we know now is that these approaches have altogether been very successful in animal models of chronic liver diseases, but we have no clinical translation to patients yet. So we have to ask why, when we, in all models where we use TGF-beta inhibitors that can reduce fibrosis and liver damage, why we cannot translate that to the patients. The reason for that is we have a context-related variation of the downstream signaling and the outcome. And if we think that the chronic liver disease develops over decades, then it's really hard to decide what we do when we use in a window, we use an inhibitor of the TGF-beta signaling pathway without monitoring how it acts on the different cell types that we have in the disease setting at that stage. So, and to give you a bit more feeling about the complexity, this shows, and this is already 10 years old now, this scheme, the so-called TGF-beta pathway interactome. So all these are proteins that have been described, and now there are maybe at least double that much proteins that have been described to somehow have a regulatory and modulating effect at the different levels of signaling from the cell surface to the nucleus. And so you can imagine, to get a clear picture at each stage of a disease that develops over decades is rather complicated. So Loredana Ciuclan, she was a student in my lab. She made her PhD and she investigated the TGF-beta signaling pathway in early stages of alcoholic liver disease. And she could show by using an antibody against the phosphorylated form of SMAD2, which can be used in immunohistochemistry or immunofluorescent investigations, as well as in immunoblotting investigation. And she has tested this antibody and looked for SMAD activation in ethanol-fed mice. We have done this in collaboration with Hide Tsukamoto, and he is using this intragastric feeding model of alcohol. And you can easily see that after 6 weeks of ethanol treatment, you have a significant steatosis. And you can also see that there's a lot of red staining in the nuclei. That means we have, in very early disease stages, we have a very strong TGF-beta signaling available. This can also be shown in immunoblots from the tissue. Phosphosmad2, phosphosmad1, 3 are activated in the tissue. This was also quantified here. And in collaboration with Helmut Seitz, a clinician who has been working in alcoholic liver disease since his life. So we have stained also patients with alcoholic liver disease for the phosphosmad activation. And you can also see that in human ASH patients, we have a similar situation like what we have found here in the mice. Just to show you a little bit in a higher magnification, that it's not only the hepatocytes with the large nuclei that show signaling, we also have that in the sinusoids where the stellate cells, the endothelial cells, and also macrophages are located. So we can say that the TGF-beta signaling system is activated quite early in alcoholic liver disease. And in this collaboration with Seitz, we have shown that the etiology, it's not in each of the liver diseases that TGF-beta is a major driver. We have also some where IL-13 is more dominant. But we could say from this investigation that alcoholic liver disease is one of those that is driven by TGF-beta signaling. Then the Loredana, I showed you before, she finished her PhD and we published that paper where she shows that by investigations mainly with primary hepatocytes from mice, that TGF-beta enhances alcohol-dependent hepatocyte damage via downregulation of alcohol dehydrogenase 1. You see here in this treatment approach that by using TGF-beta and alcohol, that the damaging effect of alcohol is enhanced by TGF-beta. Then you can see here the double treatment. You see also that with looking at antioxidants reactions, that also here the effect is enhanced if we treat the cells with both. Lipid peroxidation is significantly increased in this setting, and we have more oxidative stress if we have TGF-beta and alcohol at the same time around. When we look for the TGF-beta expression profile in the hepatocytes, so we treated cultured hepatocytes with TGF-beta for 24 hours, and then looked here especially for the metabolic genes, and you see that most of the hepatocyte metabolic genes are downregulated by TGF-beta, which shows you already that in this setting, we have this effect that goes into transdifferentiation, loss of function, myofibroblasts, or let's say tumorigenic mesenchymal transdifferentiation of the cells. And what was also obvious here is that alcohol dehydrogenase 1 is one of the mostly and strongest regulated targets here. So this has been then investigated in more detail. You see here the expression at the RNA level, dose-dependent downregulation. Looking at the ADH activity. This is a compound inhibitor and it's an inducer of the activity to show that the assay is working in principle. So activity and expression is decreased by TGF-beta, also at the protein level shown here, and also in the animal model where we make let's say a binge challenge with alcohol. You can see that immediately ADH1 expression is decreased by TGF-beta. No, this is this was the TGF-beta transgenic mice where we have overexpression in the liver of active TGF-beta, and you can see here that also in the mice, without any other challenge, the ADH1 expression is decreased. So we want to end up with the scheme that you might know, that in principle, if you have normal amounts of alcohol, this is mainly metabolized by the ADH1 system and a little bit by the so-called Mayer system using CYP2E1. And then we have an efficient alcohol metabolite excretion. And in a setting where we have excess or chronic alcohol consumption, the homeostasis is lost and it's going more this branch where we have enhanced Meus and CYP2E1 dependent metabolism. And then we have all these components that finally lead to liver damage, fat accumulation, oxidative stress. And if you have TGF-beta around, this ADH1 branch is even more inhibited. And then this part that is damaging to the liver is further enhanced. So just recently, we have published this paper in Cell Death and Disease, where we show ethanol sensitizes hepatocytes for TGF-beta triggered apoptosis. And now I come back to that scheme I showed you before, that in principle, TGF-beta has a cytostatic effect, and what I mentioned also this morning in our discussion, that everything is somehow integrating around survival pathways. And what we have investigated here is, like I showed you before, we have done the treatment, single and double treatment with TGF-beta and ethanol. And you see here that apoptosis in the caspase-3 assay and also here in caspase-3 immunoblotting, you see there's this very significant effect on hepatocyte cell death when we have TGF-beta and ethanol around in the setting of a hepatocyte monolayer culture. This is important. So you see here in the control, we have some activation of AKT. If we have ethanol, then the AKT activation is decreased. TGF-beta alone also is further inducing phospho-AKT in the setting of hepatocytes culture. And if we have both, then AKT is decreased, and this is based on ethanol. So ethanol is leading to a decrease in AKT activation. So we can also show that ethanol is inducing PTEN. You know that PTEN is a component of the beta-catenin GSK3 beta pathway, and that means it's an inhibitor, so that interferes with survival. So you see that ethanol by itself is inducing this pathway by upregulating PTEN. We also showed here in this assay, when we inhibit GSK3 beta, then the pro-apoptotic ethanol effect is rescued. We use an inhibitor for this enzyme, and that leads then to a rescue of this upregulation with regard to apoptosis. That means that alcohol somehow uses this P10 GSK3 beta system to sensitize the hepatocytes for cell death. And that this is BCL2 dependent, we show here with adenovirus experiments. You see the effect of TGF-beta and TGF-beta and ethanol here. But if we express BCL2, then we can completely rescue this effect. And finally, what is also interesting is we have used this system in some patient samples where we dissect tumors from HCC patients and perform this tissue slice culture of this, and we treated both then with TGF-beta and ethanol, and also here we can, by the double treatment, we can induce cell death in these hepatocellular carcinoma patient samples. We also show that ethanol by itself is inducing the TGF-beta system, so it enhances the expression of the type 2 receptor. Yeah, this is in vitro in the cultured hepatocytes, but also in the mice, where we have this experiment where we do the ethanol binge, and you can see that with ethanol, TGF-beta receptor 2 expression in the liver is upregulated. This can also be measured at the protein or phospho-SMAD activation level. You see here, this is TGF-beta, this is TGF-beta, and pretreatment with ethanol. You see a significant difference in the two settings. Yeah, this is a TGF-beta signaling reporter that we use in the cells, although this one is sensitized by pretreatment of the cells with TGF-beta, with ethanol. So that means somehow the TGF-beta sensitivity of the cells is as well enhanced. Also, this is a TGF-beta target genes, CTGF, which is induced by TGF-beta, and the induction is enhanced by the double treatment. And also the same can be seen for typical target genes of the TGF-beta signaling pathway, SMAD7, the negative regulator, and also SMAD6. You see here the ethanol situation, you see the TGF-beta situation, and you see the TGF-beta plus ethanol situation. So very consistent. We can conclude that these two things are induced by alcohol. Then we investigated if alcohol metabolism does play a role. So you can see here we have added oxidative stress instead of alcohol. We get the same result. Yeah. Then we have in this experiment we use N-acetylcysteine as a stress inhibitor. So you can see that this N-acetylcysteine does not have any effect by using TGF-beta and ethanol together. Yeah, you see here in this experiment that acetaldehyde, what was also very interesting finding, acetaldehyde, the metabolite of ethanol, which has a lot of other damaging and tremendous effects, tremendous adverse effects, could not phenocopy the effect of alcohol. And we could also show that by interfering with the alcohol metabolism, we couldn't show any effect here. So we use DAS and 4-MP to interfere with alcohol metabolism. And you can see that the effect of induction by TGF-beta and ethanol could not be rescued. So we have this summary scheme shown here. This shows the people here, Katja Breitkopf and Christoph Meyer, who mainly performed these experiments. You can see that we have a TGF-beta pathway that is going to apoptosis using an intrinsic mitochondrial pathway. And these are the components that are upregulated downstream of TGF-beta, and these are the components that are downregulated. Then you see that when we have ethanol, the CYP2E1 is upregulated, and this pathway is then reactivated and interferes with AKT signaling. And that means that if we have ethanol, this apoptotic route is enhanced. I come back to that situation to make the story a bit more clear. This shows you what we normally have when we culture the cells in the monolayer situation. We have discussed about that. When you have the primary hepatocytes and put it to culture, they lose the phenotype of hepatocytes very fast. So that means they have culture stress, and this culture stress, they upregulate these survival signaling pathways. And you see here, if we treat the cells in this setting with TGF-beta, then this is even enhanced. We have some kind of EMT. Yeah, we upregulate markers like vimentin and so on. That means in this setting of culture, we have this survival activation, stress kinase survival activation. And this leads then to insensitivity to the TGF-beta mediated apoptotic or cytostatic branch. Now, this is shown here again in the scheme. So this is when we have normal and healthy hepatocytes. When we have TGF-beta, they undergo apoptosis. So in a setting of culture stress, we have, for example, ECM, extracellular matrix stiffness, growth factors around. Then these signaling pathways are activated by the stress of the hepatocytes in this setting. So we have PI3K, AKT activation, we have ERK activation, and we have Wnt beta-catenin activation, and this leads to survival and proliferative activity of the hepatocytes. And in this setting, the TGF-beta pathway that is going to apoptosis is inhibited. And this is mainly mediated via P-phospho-AKT, and also the Chun kinase is somehow integrated here. In this setting, then TGF-beta itself is then inducing all these tumorigenic actions as shown here. Via caveolin, it's further inducing AKT signaling. So via pathways that the SMAD-dependent pathways, all these players here that are related to EMT, to loss of epithelial character, there are these transcription factors that induce EMT. We have these mesenchymal markers. We have downregulation of these epithelial markers. Then we have the expression of components that feed the surrounding, that are secreted in the cells, like VEGF, CTGF, TGF-beta itself, PDGF. All these components are then secreted from the hepatocytes and then feed, when we think about a more complex situation with stroma around, they feed then the tumor surrounding. And this is the way how TGF-beta is then facilitating tumor development. I think that that's work that we have performed in the last 10 years, just summarized in this scheme. Honglei Wang and Patricia Godoy and Yan Liu were those people that have been heavily involved, but also collaborations with Isabel Fabregat and people from the IT Liver Network have been participating in that. Shows you then that this situation, at least in the setting of the hepatocyte culture as a monolayer, is reversible. So if we use alcohol, as I showed you, we can reactivate this pro-apoptotic branch here. And we can use inhibitors for AKT and we get the same result. Yeah. And so that means somehow we can study here how we might interfere also with that in patients. So in collaboration with Kaigo Machida and Hide Tsukamoto, I will tell you another story that also integrates to the TGF-beta signaling pathway and loss of cytostatic TGF-beta signaling, which then ends up in cancer and especially also in alcoholic liver disease related cancer. And this is by asking the question how HCV and alcohol play together to facilitate cancer development, HCC development. So this is shown here. In the first study, Keigo investigated what happens in mice when they are infected with HCV. And what he could show is that HCV infection induces TLR4 expression in the hepatocytes. And with that, when on top of that, you feed alcohol to the animals, then LPS can directly bind to TLR4 on the surface of the hepatocytes. And this led then to the expression of NANOC and the formation of liver cancer. And molecularly, this NANOC is a stemness factor. So that means that in this regard, we have the generation of cancer stem cells in the liver. And molecularly, what he did, he isolated these cancer stem cells and the tumors from the liver, also from human patients, and then made a library and found out that there were some oncogenes involved that were downstream positively regulated by NANOC. This NANOC induced IGF-2, BP3, and JAP1. And with many experiments, he found out that this IGF-BP3 and also JAP1 integrate to the TGF-beta signaling pathway and are responsible then for the chemo resistance that is downstream of this cancer stem cell formation. So IGF-BP3 is integrating via AKT and mTOR signaling, and with this inhibits the cytostatic downstream signaling of the SMAD3 protein. So there is formed a complex of these two, and the binding of the cytostatic target genes is inhibited. And secondly, this JAP1 is a stabilizer of this biological inhibitor of TGF-beta, SMAD7, is the negative feedback loop. TGF-beta is inducing SMAD7. SMAD7 is binding to the receptor complex and then degrading the receptor complex. And in this setting, JAK1 is a stabilizer of SMAD7. And then we have, from two sides, we have an inhibition of the TGF-beta downstream signaling. And in approaches using shRNAs targeting the two oncogenes, this setting could be rescued, and we could make the cells again sensitive to chemotherapy, and could show by this how HCV and alcohol play together to facilitate liver cancer formation. This slide is highly busy. I just wanted to show you a component of these signaling pathways. I even don't see it here right now. There must be somewhere TAC1. Yeah, and Akihiro Seiki, he published a paper which I found very interesting. So he used this TAC1, which is like this AKT I told you before, is a survival signaling pathway, and at least is a part of NF-κB survival signaling. And he made a knockout mouse for TAC1. And these mice displayed a very severe phenotype. Within 3 months, they developed very severe liver cancer. And interestingly, when he used a double knockout mouse where he depleted, in addition, the TGF-beta type 2 receptor in the mice, then the phenotype was completely rescued. Why I show you this here? So this shows you just how it is. You see very severe damage after 4 weeks in the mice. You have a very strong inflammation here, the macrophages, and you always hear in the number 4 situation, you have the TGF-beta receptor knockout, double knockout situation. You see the complete rescue always. Inflammation, number of macrophages, and then apoptosis and compensatory proliferation. I think this is very interesting, and this also tells you something about the role of inflammation in cancer formation. Because you see here that you have a very severe damage and apoptosis going on, which is due to the loss of survival signaling in the hepatocytes, and you don't have a spontaneous damage, but the TGF-beta that is around in the system is sufficient to induce such a massive cell death in these hepatocytes that you have a compensatory proliferative activity that finally leads then to mistakes and to cancer formation. Okay, this shows you the summary slide integrating all this information again. So we have TGF-beta, we have hepatocytes under cell stress, which activates survival signaling pathways, also NF-κB included now. And then we have the tumor suppressor function of TGF-beta. You see here the apoptotic route. And when the hepatocytes are under cell stress, these players that induce survival, they interfere with that branch. TGF-beta is then going another way, and that is shown here. It's inducing growth factors, it's inducing growth factor receptors, it's activating survival signaling pathways by itself, it's inducing EMT. We have this setting with NANOG and JAP IGF-BP2 that also inhibit the cytostatic branch, and all these actions interfere with the cytostatic pathway, and this can be reversed, for example, by inhibiting AKT, so by interfering with survival signaling. But it also shows you that we should not do that in a very dramatic way, because then we can induce cancer formation by just having a too strong cytostatic effect. Okay, coming back to that slide, you remember that I showed you very early in this alcoholic liver disease setting, we have TGF-beta around. So we put the question now, where does the activated TGF-beta come from in this setting? So I give you some biological information about TGF-beta. So TGF-beta is stored in an inactive form in the extracellular matrix. So this is shown here. So we have, after synthesis from the genes, TGF-beta is transported and secreted in a form. So here, this is the cytokine, and then we have latency-associated peptide, and we have the latent TGF-beta binding protein. And as such a complex TGF-beta is deposited in the extracellular matrix in this latent form. So it's quiescent. And then it can be activated by integrins. And then the TGF-beta is set free and binds then to the receptor and initiates the signaling. And this ends then in what we know, the activation of stellate cells, for example. So then I want to report now in the second part of my talk about this protein ECM1, which we defined as a gatekeeper that restrains TGF-beta activation to maintain liver homeostasis and prevent fibrogenesis. This is a collaboration between our institute and the Pasteur Institute in Shanghai and some hospitals in China, and the PhD student who is now a postdoc. He finished his PhD with that work, Waigu Fan. I show him here. He just presented the results at the ASLD recently, so the poster was selected to a talk, and it's currently in investigation. So first, what is extracellular matrix protein 1? It localizes in the extracellular matrix of a normal liver. This is what we found out. This is the protein structure as shown here. Maybe not so important at that time. What was known about it? It's a secreted glycoprotein. There are some papers showing that it is overexpressed in cancer and thereby may facilitate proliferation. More importantly, mutation in these proteins are dedicated to a very rare disease. It's called lipoid proteinosis. It's something that also makes you deposits in the brain cells, and the people are depressive, and they cannot somehow they cannot analyze from you how you look, what you feel. That's a deficiency here, and they have also skin problems. And in a recent paper of this group, and this is how we came together, because actually the group of Waigu, they are immunologists and they are not liver experts, and they have been investigating this ECM1 protein, and they found out that it has some effect on the differentiation of T cells, and they also found out that this is related to TGF-beta somehow, and that brought us together. And so we have investigated in the liver that, or let's say in a organ plot, we could show that the liver and the kidney are those organs that mostly express the protein, and we could also show by immunostainings that it is deposited in the extracellular matrix in the liver. And then we have used the patients collect cohort of Ramon Batalla. He has published array data, and there is in the near future he will publish much more patient samples, RNA-seq data, and so we found that this ECM1 protein is significantly decreased in patients with alcoholic hepatitis. This you could also show by the immunohistochemical staining. So we were asking about what about ECM1 in other fibrosis models, and so we used the CCL4 model, a NASH model, and the BDL model. You can easily see that in all these models, ECM1 is significantly downregulated in the disease setting, also visible at the immunoblood levels. Then they developed knockout mice, and these knockout mice have a very severe phenotype. You see that nearly all mice are dead until the age of 10 weeks, and these mice display cirrhotic livers. They have ascites that can be collected, and this shows you the histology. You see a very strongly disturbed liver architecture here with very severe fibrosis, stellate cell activation here. You see here the hydroxyproline content in the livers, alpha-SMA expression, collagen expression, type 1, type 3, desmin, number of stellate cells. Also immunohistochemical stainings. Glutamine synthetase shows you that the zonation of the liver is severely disturbed in the knockout mice. You see a significant ductular reaction here with the CK19 staining, CD26 inflammation. The ICAM1 staining shows you that the vessel system is also severely disturbed. So we have very significant disturbance of the liver architecture in the knockout mice. So then we were asking which cell type is producing ECM1. And here we have used the CCL4 model and control mice and have isolated the different cell types. And you can see that all the cells express to some extent this protein, but mainly the hepatocytes are responsible for ECM1 expression. And if we have a disease setting, the hepatocytes downregulate the expression of that protein. So when we ask how does ECM1 affect liver fibrosis, so when we look in the tissue about damage, so you see there is in the knockout mice, even if we have such a severe fibrosis, we do not have a lot of apoptosis as compared to the APAP situation, for example, here. Many apoptotic cells, here we don't find that. Or when we look for inflammation, here the number of macrophages, also here as compared to the MDR2 knockout mice or 6-week CCL4, you even have not so much inflammation available. So we look for TGF-beta expression. So we don't see a big difference in the TGF-beta at the mRNA level. But when you look for SMAD activation, you see a very significant activation of SMAD3 phosphorylation here. You see a very strong signal. And this we could also phenocopy at the histochemical level by using the phosphosmart antibody. Yeah, so coming back to that situation, the TGF-beta receptor, TGF-beta ligand is produced and deposited in the extracellular matrix. So maybe the ECM1 protein is related to this extracellular matrix deposited TGF-beta and has some effect in this quiescence. So then we know that these integrin proteins are able to activate the latent TGF-beta. So we performed co-IP experiments to show that ECM1 and integrins might be forming complexes. And this we could show here by using integrin or using ECM1 as target. We could show that we could immunoprecipitate the other partner in both experimental settings. And we also performed the immuno co-staining with fluorescence microscopy. You can see that ECM1 and integrin alpha-5 are co-localized in the mice. We also performed functional assays. This is a culture system where we use TGF-beta reporter cells. They have a promoter for the SMADs, for SMAD binding integrated, and then these are used and they secrete then luciferase into the medium, which can be measured. And we perform a co-culture experiment with stellate cells and investigated then the effect of ECM1. And also here we could show that by using ECM1 as a recombinant protein, like when we use this RGD motif from the integrin, we could inhibit the TGF-beta activation in the supernatant, which functionally confirms that ECM1 is indeed involved in the activation of latent TGF-beta. And this also reflects then in the parameters that lead to stellate cell activation, alpha-smooth muscle actin, and type 1 collagen. So in the next step, we try to confirm that in the in vivo setting. So we used an AAV virus, which expresses a soluble type 2 receptor. It's a similar principle like having these flocks or these knockout mice for the type 2 receptor. And if you overexpress this receptor in the tissue, then the ligand that is activated will be catched and will not go for signaling. And this is shown here. So we have the control and TGF-beta receptor AAV virus, and you see that the phosphosmad3 could be decreased in the mice. And what we found is we could completely rescue the mice from at least from mortality and from death, as shown here. And this you can also see in the tissue. Yeah, so with a soluble type 2 TGF-beta receptor, we could completely rescue this phenotype of severe stellate cell activation and also matrix deposition, which means that TGF-beta is sufficient to inhibit the severe effect of ECM1 down regulation. That means that all what we see is somehow related to TGF-beta. So asking the question if ECM1 can work as a therapeutic agent. So when we look in patient tissue, we have done in a cohort of patients with different disease stages based on immunohistochemical stainings and quantification, we show that indeed with severity, the expression of ECM1 is decreasing in the patient. These are HBV patients that have been investigated in China. Yeah, and you can see that also if we overexpress ECM1 in the model of CCL4, we can also rescue the phenotype. You see here, we use AAV ECM1 and infect the mice. And you see here with the flag tag detection, you see the overexpression of ECM1 in these mice, and we could rescue the hydroxyproline content in the mice, and also from the stereous red staining, you could see that we could inhibit or interfere with CCL4-induced liver damage. This is also then in the knockout mice, we overexpress ECM1 by delivery of an AAV, which is shown here. And you also can see that we could, by delivering ECM1 back into the system, we could rescue mortality of the mice, we could decrease hydroxyproline content, and we could also restore a relatively normal liver architecture. Shown here, what can we say now? This is the graphical abstract, more or less. We have the healthy situation with the latent TGF-beta, with latency binding protein ECM1 integrated now in this complex. And in the setting of liver disease, this ECM1 is decreased, is not available anymore in higher concentrations. And with that, TGF-beta, which is in the latent form, will be activated and will start to drive the fibrogenic process. So what will we learn from that? The physiology, so we have learned from that there is obviously a huge amount of latent TGF-beta deposited in the healthy liver. From the pathomechanism, I think this ECM1 protein seems to be some kind of damage-associated signal. That means the hepatocytes under stress, they might downregulate ECM1 expression, which then starts to trigger the fibrotic process with HCV activation and fibrogenesis. And I think also with regard to therapy, this ECM1 peptide might be promising because this protein is in principle present in the liver anyway, and it's decreased. So in a disease setting, you have reduced amounts. So by using a peptide, you might not expect too many side effects. I want to mention our funding, Liver Systems Medicine, a network of the federal government in Germany. It's about systems biology, and the DFG, DAAD, and this is over. This was a Marie Curie International Training Network. It's over, but some of the data I presented were still funded by that. And with that, I thank you for your attention. This is very fast because they already do that intrinsically. If you don't do anything, they have this culture stress.
All right.
Yeah. So you immediately have EMT. So it's even the other way around. You have to start very early that you still can measure an effect of TGF-beta, that some of the cells remain in the more epithelial phenotype. I have learned that the human hepatocytes, they are more stable. So you have more time. So you can still see the structure of the hepatocytes with polarity during the course of a week or 10 days even. And you can get into such setting if I mentioned that before, when you use such a collagen sandwich culture system. So you have the feeling when the hepatocytes think there's a three-dimensional structure, they feel a little bit more healthy and they keep their shape a little longer. These mice die already after 10 weeks. So we have to think about when we we use maybe the mice in a heterogeneous, in a heterozygous setting, that we can investigate how they respond to the induction of tumors. Very good question. And actually, I exactly wanted to bring you to that point, that on one hand, you want to have that, of course, because you have damage and you need to repair. But I think it's also a matter of the severity of damage. When you have too many cells dead and the compensatory system is overactivated, then the chance to that mistakes will manifest is very high. And this is exactly what happens there. You have such a strong compensatory proliferation. Many cells proliferate unusually much. You see, nearly every second hepatocyte was positive for Ki67 in this setting. And then you have tumors within 3 months. You have massive tumors in the liver. That shows you clearly, if you have too much apoptosis and then too much proliferation, that cannot be good. This is also dependent on what you want to investigate. If you want to investigate the route from alcohol to steatosis, then you can rely on Lieber-DeCarli. Yeah, that will be fine. Fibrosis, I think the best model for fibrosis, the Tsukamoto model, is maybe even in Germany, the ethics committee would not agree on doing that. Feeding the mice with a pump intragastric with alcohol every day. I think this NIAAA model is promising if you use it in more cycles than only one. Yeah, so if you have a chronic feeding, then binge. And then again, chronic and binge. So have several binges, several chronic feeding, which is actually also something that is the real life situation. But on the other hand, I mentioned also that currently many labs are on the way to try to establish a model that at least to some extent phenocopy the situation in these more severe diseased patients. That means when they have this acute on chronic liver failure situation where you have a compensated cirrhosis and then suddenly they come to the hospital and they have a very short mortality, very high mortality within 30 days, and which have this golden window where some of them recover. So that I think that is something we are also interested in. And I mentioned that we are using this MDR2 knockout mouse model and bring the mice already to some kind of cirrhotic stage. Also, a colleague of mine, Jan Hengsler, you know the name. They have 12 months fed CCL4 twice a week for nearly a year. And these mice have really cirrhosis. And then using this model and then on top give an alcohol binge to find out what's happening now. But it's very complicated. You have this infection situation, and maybe here your model might be interesting that you mentioned that where you have a higher infection rate. Maybe that has something to do with the shape of the gut. All the kind of drugs, we have not tested that. But we could phenocopy it by using oxidative stress, by increasing oxidative stress. So oxidative stress is definitely downstream of ethanol here. But what we also could show, it's not the metabolism. We could inhibit the metabolism and we could not inhibit the effect of alcohol. That means it seems to be the alcohol molecule itself has some effect here. And with regard to transcriptional activity, I have to say that the data that we have now, it's indirect data. That means it's 24 hours, 48 hours after treatment. That means we cannot say that there is a direct route from ethanol to the promoters of the genes. would be very interesting. We want to investigate that, especially with regard to the upregulation of the type 2 receptor expression, which is very interesting to us. So alcohol is inducing TGF-beta type 2 receptor at the mRNA level. I think I would expect that the soluble TGF-beta type 2 receptor is more efficient than the compound, because compounds are more diluted. Here you have a protein that is present. You can measure in the tissue that the protein is there. And actually, this has been used long time ago already, the soluble receptor with adenoviruses.
I know.
I think...
It was used even before than the kinase inhibitors, then we moved to the kinase inhibitors. Mm-hmm.
And I would expect more specificity with that compound or with that tool as compared to, but you cannot use it in patients, I think. So I think there are no data available on that. It would be very interesting to investigate that with a tool looking for phosphosmell. Very interesting, but I don't know. We don't have data. One argument against that would be that it is deposited in the matrix in a latent form, which is also known for other cytokines like HGF, which maybe the tissue needs very fast if there is a situation of damage or alarming situation, that it might be activated without synthesis. And so but I have shown you that there are multiple levels of regulation. So why not? TGF-beta could be transported by vesicles also. I think if it's let's say in a cell and a vesicle is formed, I'm not sure if the vesicle formation is really targeting specific molecules. I think what is there will be taken, more or less. That is, at least to some extent, what I would expect.
Thanks. No more questions from Barcelona. Thank you, Stephen.
Okay, thank you very much, Barcelona, and thanks to Dr. Dolly for this very nice talk, and see you very soon in the liver seminars.